Anytime where you are considering testing how a protein, such as an RNA polymerase, is going to bind to a nucleic acid, such as a double-stranded piece of DNA, a convenient way to test for that is a filter binding assay. The principle of a filter binding assay is that you take a nitrocellulose filter, whereas you first have your solution of double-stranded DNA, and that alone should bind very poorly to your nitrocellulose filter, and it all should fall out. Whereas protein, however, can survive very nicely on nitrocellulose, and it's retained by the filter. Therefore, if you want to test whether a protein binds to a strand of DNA, you have both of them in your Eppendorf tube. If now your DNA is retained by the filter, then you can assume that it is bound to the protein. And you can measure this amount of DNA that's bound. Most of the time, you radio-labeled your DNA first off, so that if any of the radio label gets tracked on your filter, then you take a little piece of that filter with tweezers and you put it in a scintillation vial and count the radioactivity. Just remember, all of these experiments are done in comparison to a control. So you would have one tube with your DNA alone and another tube with DNA with protein. This is a convenient way to test if protein binds to DNA.